Welcome to the last day of MetPhys 2020, day 29, where we're going to hear from Ramon, Klein, Geltink about his fantastic work. I've said this every day, and I'll say it one last time. MetPhys 2020 has ushered in a new model for inclusive global science. We're able to put together some fantastic talks up there with keystone level speakers that we've been able to distribute globally by making it free on YouTube, so anyone can listen to them anywhere at any time. And we're happy to take the cost of that with the White Magara Lab. So it's a very cost-effective means. It's also great for the planet because we are not all flying to get to one destination to listen to these talks. So we hope that this model continues, and we've been glad to see other seminar series take on the model of making their talks available on YouTube so anyone can listen to them at their own leisure. Cassidy took on the Inception Challenge from MetPhys 2020. This blew my mind that she had this devotion to take a photo every day for the entire series in front of the computer. And what she put together was an amazing cluster of photos. So we're going to go through them right now. Blink and you'll miss it. But thank you, Cassidy. Well done. Congratulations. You are the MetPhys 2020 Inception champion. And here we go. If you look closely, you'll see those teeth recede back into her gums. That's 29 days of photos, folks. Whoa. And like that, it's over. So there are some more prizes to hand out. Yesterday, we finished the poll on who had the best cup. There were many cups that entered. This was the most hotly contested competition. I whittled it down to 4. I know I upset some people with my choices. It was very close yesterday when I put it up. Actually, Thierry Aki's Neuron homemade cup was in the lead. However, with a little bit of lobbying, Asma El Kanawi was able to take over and actually take home that prize. She is now the proud recipient of the MetPhys 2020 prize for the best mug. And I was pretty impressed by the other competitors, Anna Miguel and Thierry, coming in early, conceding the defeat, and complaining about the reviewers, which is the right way to do things. We had an earlier poll with Isaac Harris. I just want to reiterate, he definitely took home the prize with his cat meme. Thank you to the other entrants. This was kind of fantastic. And I know today's speaker would have loved to be in the challenge. I think he would have won. And we're going to enjoy his Zoom background today. Felix, you've taken home the MetPhys 2020 prize for most curve flattened. Not only did you cycle while listed in MetPhys 2020, you also shared the data, which is the only scientific way to do it. And that data was shared immediately upon request. That is open science, people. That is sharing your raw data to prove that you are not making it up. Thank you, Felix. He won the prize for the most curves flattened. John Campbell took home the prize for the most kids fattened. I love his Pac-Man game that he's using with the treadmill. If I had a treadmill, I would be doing that with my kids. It works less well on the cycle. We kind of spin the pedal and they fly off, and that's not very happy for the kids, but hey, the cats are happy. Miriam Valera Alberti came in with a suggestion for a new kind of coffee I'd not heard about, and she has won the MetPhys 2020 prize for a drink suggestion that has made me the most fattened. And those Dalgona coffees are keeping me happy every afternoon. Thank you, Miriam. Gareth Lim has taken home the prize for sticking to your guns and instead making buns. We challenged him to make latte art. He said, I do not mix coffee with milk. I'm standing up to that. I'm not going to do it. But he instead baked a ton of buns. And so for sticking to your guns and making buns, Gareth, you've won a MetPhys 2020 prize. I was going to run a prize for the very best good boy or girl. It turns out that you just can't pick. They're all the very best good boy or girl. And congratulations to all the dogs and your entrants. I was also going to run a prize for the cats, but it turns out they just don't care. So no cat prizes here. And I was going to run a prize for the best children. However, it doesn't seem like we could really pick one. So thank you for all your children's participation and taking photos. I know that lit up my day. So thank you all for listening at home and your participation. This has been a truly fun experience for me, and I hope it has been for you all at home. As we go forward and we're not having the talks, if you still want to share with the community, feel free to use that hashtag. And I look forward to more shares in MetPhys 2021 next year. None of this would have been possible without the fantastic speakers who've not only been kind and agreed very rapidly to give a talk, they've also been open with their data, sharing unpublished work and showing that even if they suspect there's going to be a broad audience listening to them on the internet, they're not going to let the scoop monster get into their heads, and they are going to be free and open to share their data, which is, I think, one of the main concerns about having talks online was, will the speakers even present anything that we care about? And in fact, we've been able to show that they will, and I'm really glad that they've chosen to make that strong impact on the community. Our talks went out for 2 weeks more, and I thank the additional speakers for joining in. I know that we've all enjoyed these last 2 weeks of talks. They've been just as great as the first 4, and I'm really grateful for those speakers for agreeing to give a talk. And I'm looking forward to the speakers for next year. I know we've already got a fantastic lineup, and I'll probably release that towards the end of the year. If you'd like to thank me, the way to do that is to, if you have the means, reach out to the Pathway to Stop Diabetes program from the American Diabetes Association. I know my lab wouldn't be in the situation it's in without their generous support, and they're not just supporting me, they're supporting a large number of fantastic projects. And if you go on the website, you can read all about the progress being made and the projects being funded. Of course, to keep funding fantastic scientists from our community, they do need additional support. So share this with your network, with people who are interested, who say, I'd like to help moving the research forward. How can I do that? Let them know that this is a one path to do that. So without further ado, I had promised you that we will take care of this lift. I will never make a promise on Twitter that I don't keep, or I might, but this one's not. So without further ado, I am going to stop my share and see if we can nail it. They said nobody puts baby in a corner. I'm just hoping not to put my wife in a box. All right, so. Here we go. And we have music. Are you ready? They're ready. Come on, let's do this. And that's as good as we're going to get, folks. Thank you for listening. All right, Ramon, I'm transferring over to you now, man. It's up to you. All right, Ramon, I'm going to make you. Thank you, everyone, for listening at home. Bye. See you all in the Twitter space. Ramon, we can see you.
Okay, share my screen.
Screen is coming up, waiting for it to go full. And it looks great. We can hear you. Have a nice talk. See you all on Twitter.
Perfect. Thanks, Philip. So let me start by thanking the organizers for the invite to give a talk in this amazing seminar series, MetPhys 2020. It's been very impressive. I'm kind of daunted by the fact that I'm the last speaker in this series. Mentions of Keystone-like speakers. I hope I can live up to the challenge here. So the talk I want to give today is about glucose metabolism in CD8 T cells, and mostly about conditioning a tumor-killing army. And this title is kind of provocative, but I hope to get to why we chose this title for the talk. I want to highlight again, as Phil said, questions will be answered through Twitter. So don't use the raise hand function. Just put something on Twitter, tag me or tag MetPhys 2020. Joy is online too, my co-first author for the story that I'm presenting from Erica's lab. So we're happy to answer any questions. Looking forward to the interactions. So first, seeing as Phil didn't take the time to introduce the cats, I want to at least highlight one of them. So Maya has been in our house part of the whole seminar series. Pretty much every day, she's sitting there and actually staring. This is not fabricated. She actually does this. So this became kind of a breakfast ritual. And given that this is the last talk today, we're really going to miss that. It kind of gave us some normalcy in isolation. So it's kind of tough. And especially for Cassidy, since she mentioned there should be a cat-focused one, she also photobombed one of the Inception Challenge photos. So that takes me back to all this shenanigans. First, I want to say, since it's April 24th, happy birthday to my dad, who's locked in his house at home together with my mom. Try not to get COVID-19. So keep going, stay strong, and enjoy your birthday drinks, since it's the afternoon in Holland right now. And then we'll carry on with the actual science. So when I talk about CD8 T cells and conditioning a tumor killing army, the end of the talk will highlight this figure here. And I just want to highlight this in the beginning, because this is really what I want to work towards. So we really want to be able to use these CD8 T cells, which are the effector cells of the immune system, to attack tumors and to get better at it. And there have been some talks already by Thales, by Alex Muir, who highlighted like tumor microenvironment challenges and stuff. These T cells in the tumor microenvironment are also challenged, and they don't always manage to contain tumor growth. So when we do an experiment like this, where we give mice flank tumors at day 0, let them grow for 8 days, and then inject cells that are specifically attacking these tumors, and we'll get back to this later in the talk, what exactly these do and what they are. We get 2 phenotypes, one here in orange, that clears the tumor growth. So it completely gets rid of these tumors and they don't come back. And the ones in black, which were thought to be the best effector cells we can make in a culturing dish, or the like standard activation, they fail to control the tumor growth in half these conditions. And you unfortunately have to sacrifice these mice because these tumors get rather big. So what is a CD8 T cell? So one of the things I wanted to highlight, and this kind of came back to me in the last couple of days as I was preparing this talk, I don't know if anybody's familiar with this series. It's a French cartoon series from the 80s, which is called Once Upon a Time Life. There were 3 series of it. The Life one is all about the body, and they actually accurately depicted the T cells as these floating machines with a gun on the front that go through the circulation, through tissues, find intruders like viruses, bacteria, and tumors, and they kill them with this really fancy blue smoke. So I definitely recommend you to look this up. It's in French, but dubbed in multiple different languages. So I really enjoyed this as a kid, so it's kind of funny. Also important to mention, in this program, they had it right. The T cells were the important ones. They mentioned B cells, but the rest of the lymphocytes are kind of ignored. So it's kind of funny to see that they already were talking about this kind of phenotype, and I'm going back to that right now. So the talk today, so this is the presentation overview, is kind of split into 2 parts. So the first part is about this metabolic conditioning for adoptive cellular therapy, which I hope to convince you of in the next hour or so. This is an unpublished study which we just resubmitted. And this was done in Erica Pierce's lab, so my postdoc supervisor at the Max Planck Institute in Freiburg in Germany. Erica's a fantastic mentor. If anybody's looking for a great boss, definitely look into her if she's recruiting, because it's a great place to do science. And this work was spearheaded by myself together with Joy Edwards-Hicks, who is an absolute mass spec aficionado, and she really helped push this story to where it came. So I'll highlight a lot of her work as well, since we really finished this side by side, especially for the revision, since I left last summer to start my lab here in UBC. So the Department of Pathology and Laboratory Medicine, and we're located at the BC Children's Hospital Research Institute in Vancouver. So given that we left last summer, Joy really pushed the revision experiments and made sure that we could resubmit this paper. So hopefully the reviewers are online watching this and agree that we answered most of the concerns. So part 2 is about our interests from my lab, which we just started last summer. So we don't have a whole lot of data yet that is mature enough to present here. But we're interested in cell intrinsic metabolic flexibility, and we'll get to there in the last few slides of the talk. So just to have everybody on the same page, as Phil highlighted when we were talking this morning, is there's a broad audience here, and it's physiological people, metabolic people, hardcore chemists, lots of undergrads. So I want to make sure that we're all on the same page. So first, let me define metabolism. So if you look for the definition, it says it's the chemical processes that occur within a living organism in order to maintain life, which is, I would say, rather broad. So to simplify this further, you can look at a Sigma poster like this, which is very, and you're not supposed to be able to read this. But what I just want to highlight here is that when you look at the biochemical pathways that make up carbon metabolism in a cell, as depicted here, it's incredibly complex. And the way I look at it, and this is especially important for a mixed audience, because we're looking at cellular metabolism. So we're looking at how an individual cell responds. They're dependent on metabolite availability. So if you don't have a metabolite in the surroundings, you can't use it, as highlighted by multiple people in these talks over the last couple of weeks. these metabolites don't all diffuse freely into cells. So a lot of them are not able to get in passively, so they need to be actively transporters. So you need specific metabolite transporters, which have been highlighted in multiple talks as well. And then you need the metabolic enzymes. So the metabolic enzymes are depicted in this poster as individual lines. So there's a whole lot of them, and they're incredibly interconnecting through the pathways. And then an important part of this talk is you also need metabolic cofactors. So not only do you need the enzymes and their substrates coming from metabolite availability, you need cofactors for these enzymes to function and handle these substrates. So most of the talks we've seen have divided these pathways into two things. So there's nutrients to energy with the currency of ATP, which has been highlighted by a lot of people, especially looking at glucose metabolism. On the other side, nutrients to biomass also happens. And this was actually really nicely flushed out by a bunch of different talks, but especially earlier this week by Issam Ben Sarah and Lydia Finley, who looked at the production of amino acids or nucleotides. So if you look at the example of glucose, which is highlighted here in red, you can make ATP in green through glycolysis, which we'll introduce a little bit in more detail later, or you can fully oxidize the glucose molecule and make ATP in the mitochondria down here. But I think in a lot of cases, it's overlooked that it's also used to produce biomass. So I've highlighted here in blue a couple of examples, but glucose can be used, or carbons derived from glucose can be used for a lot of different biosynthetic processes. Like purines, pyrimidines, we'll talk about nucleotides, amino acids, phospholipids, lipid species. There's a whole host of examples here. And one of the important things I want to highlight is that nutrient availability in the case of cellular metabolism, so when we're looking at T cells, just the fact that the nutrient is around doesn't mean it will be taken up by the cell. And this could be regulated by the level of metabolite transporter or choices made by the cell, if you can call it choices, so to say. And one step further, nutrient uptake doesn't necessarily mean they assimilate it into any of these biosynthetic pathways or make ATP from it either. So it could be that glucose is taken up and made into glycogen, so they're not assimilated in anything, or they can assimilate it into a whole host of different biosynthetic pathways or shed carbons in the form of lactate, which we'll get to in a little bit, without really biosynthetically benefiting the cell too much. So these are kind of the hallmarks that we want to talk about. So I'll touch upon most of these parts of the talk throughout the talk. But I want to start with one slide to introduce central carbon metabolism and in a fashion that is relevant for this talk. So I've highlighted the important pathways in different colors. So glycolysis here is in red, the TCA and OXPHOS here in blue in the mitochondria, and the pentose phosphate pathway, which we'll touch upon a bunch of times here in purple. And then we have glutathione metabolism and reduction here, which was really nicely highlighted in the setting of Ross and biosynthetic pathways by Isaac Harris and Thales as well. So when we talk about glucose metabolism or glycolysis, this is the glucose conversion to pyruvate. And in this setting, in the setting of T cells, when we talk about glycolysis, it's mostly aerobic glycolysis, where lactate is produced, which is then shed from the cell. So this generates ATP, but it's relatively inefficient. So there's 2 ATP molecules consumed here, generating ADPs. And then in the bottom part of the pathway, and I haven't spelled it all out here, but you get 4 ATPs per glucose molecule back for a net yield of 2 ATP. So it's relatively energy inefficient. The other option is to use glucose for oxidative phosphorylation. So in that case, you make pyruvate. This pyruvate is actively transported into the mitochondria, made into a 2-carbon acetyl-CoA, which then assimilates in the TCA cycle. The TCA cycle generates NADH and FADH2, and these are used in the electron transport chain in the mitochondria to produce a proton gradient from complex 1 and complex 3. There's ROS being produced, which we'll get back to, and which definitely plays a role in this pathway as well. And this ultimately leads to O2 consumption as an electron acceptor in complex 4, and ATP production by complex 5. So if you run glucose this way, it's a lactate, you get 2 ATP. If you go this route and you fully oxidize the glucose molecule, you can net up to 36 or even more ATPs, depending on how you calculate this. So this is the more energy efficient route. This is much faster. So that's kind of the separation. And it's important to mention that OXPHOS is defined as the electron transport chain conversion of NADH to ATP through the ultimate electron acceptor molecule oxygen. And it isn't always tightly linked to the turning of the TCA cycle, because this NADH can come from other places, as can FADH. But this is not necessarily important for this talk. So when we're talking about OXPHOS, we're really just measuring the activity of the electron transport chain. Then the pentose phosphate pathway, which is also branched off of glycolysis. Glucose is phosphorylated. In T cells, this happens by hexokinase. This phosphorylated glucose is then shunted this way, handled by glucose 6-phosphate dehydrogenase. A few other enzymes before you get to the pentose phosphates. And this generates a really important redox cofactor, NADPH. This NADPH is crucial in T cells to make sure that you have glutathione present. And if you lose the ability to produce NADPH, you lose the ability to revert oxidized glutathione back to glutathione, and you become sensitive to ROS. So we'll get back to that in a little bit later in the talk. But the important part of the biosynthetic angle of this, it also generates pentose phosphates, which are precursors for purines, pyrimidines, and ultimately nucleotide biosynthesis, as was highlighted earlier this week by Issam Ben Sarah. And this is not only, the nucleotides are not only coming from the pentose phosphate pathway, as he rightly indicated, there's serine synthesis involved, but these are important precursors for nucleotide biosynthesis. So these are the pathways we'll be talking about mostly. So these four that are indicated. For the non-immunologists on the call, which I'm assuming are a lot, I don't really consider myself as an immunologist either. I just want to highlight T cell metabolism in one slide. Just as a really basic background, and this is really all you need for this talk, T cells are quote unquote born in the thymus. They're either CD8 positive T cells, which we consider the effector compartment or the executors of the immune response, or the CD4 T cell compartment, which is the helper compartment, which more shapes the immune response. So when you're looking at the pro-inflammatory context, there's 3 helper subsets that are pro-inflammatory. Then you have a B cell support from the T follicular helper cells, and Tregs, which are a big focus of a lab next door, Megan Levings's lab at BC Children's, which is really pioneering Treg biology, which are anti-inflammatory cells. So we won't be talking much about the CD4 compartment today at all. We're really focusing on the CD8 subsets. And what is important is that these CD8 T cells are inherently flexible in their metabolic pathway activations. So as highlighted here, and we have 3 subtypes we'll be talking about. We're talking about naive T cells, which haven't seen antigen. Then during activation, which takes about 36 hours, you get a metabolic reprogramming, which is the focus of a lot of labs, and a lot of really elegant work has been done in this field. They become effector cells. These effector cells are the ones that execute the immune response. They live days to weeks. They're characterized by rapid proliferation and cytokine production. So these are the important ones if you were to fight a tumor based on their cytokine production and rapid proliferation. And the unique feature of these CD8 T cells is that when an infection or a tumor is resolved, they can retract into a long-lived population of memory cells. So there's many fewer of these, which live weeks to years. So these are kind of the aim of vaccination strategies. These are the cells that will protect you long-term. They undergo homeostatic proliferation, and they give you this long-lived immune protection. So this is kind of the difference between the setups. I think it's important to point out that a lot of papers are coming out now, which are much more nuanced than over the last few years. So a lot of T cell subsets utilize different metabolic programs. And I think this is becoming more and more evident, and it's much more complex than we understood it in the beginning, let's say it that way. We were habituated a little bit to label cells with metabolic phenotypes. So calling a cell glycolytic or oxidative, there's much more to it. And especially when you look at these graphs here. So many T cell subsets use at least glucose, amino acids like glutamine, serine, and they use oxygen in the mitochondria. So to just call a cell either glycolytic or oxidative is probably a vast oversimplification. So the field is really going in this direction now, where the nuance is really pulled out. So these are figures pulled from the annual reviews that we wrote in Erica's lab a few years back now. So this gets us to the model of CD8 T cell metabolism, and this is kind of important for this talk. So if you were to get infected by an extracellular pathogen or a bacterium, here I tried to represent Listeria as a pathogen. The pathogen load increases. As the pathogen load increases, your immune system recognizes that there's an issue. Antigen presenting cell will digest this pathogen, present this to a naive T cell. So the important thing here is that the antigen presentation presents it to a unique T cell receptor on a naive T cell. So you need one of these to recognize it, which then in the presence of a danger signal leads to the expansion of an effector response. So you get one of these cells expanding very rapidly into a lot of unique cells with the same receptor that can then recognize this pathogen, and by the secretion of effector molecules like interferon gamma, granzyme B, and perforin. And for this talk, interferon gamma comes up a lot, and granzyme is used a lot as a readout. And these then allow you to resolve this infection, get rid of the pathogen, and you're happy to go on, right? So this also leads, so as this resolves, you get a retraction of these large amounts of effector cells, and the proliferation dampens, some of them die off, these differentiate into memory cells, and these are the long-lived cells that last for years. And important to mention is that these still contain the same receptor. So if you were to be reinfected with the exact same pathogen, these cells will be able to recognize this much faster and mount an incredibly quick, stronger secondary effector response. So you get much more rapid expansion, and you get a more rapid resolution. So oftentimes you don't even really get sick if you have a proper memory response to one of these reinfections. So this is, as you can see here, these colors are kind of indicative of something. So these are indicative of different metabolic pathways. So as a naive cell, they're mostly dependent on oxidative metabolic pathways, depending on the mitochondrial OXPHOS and fatty acid oxidation as a naive cell. But during naive T cell activation, a lot of pathways have been shown to be important. And this has been the focus of a lot of work. And there's some examples here, and this is not by any stretch of the imagination an extensive list. There's many, many more. But notably, glutamine was shown to be very important for this by the Green and Rathmel labs, also by the Control lab, by expression of the facilitated glutamine transporter. Mitochondrial ROS has to be produced for mitochondrial complex 3, which was really nicely shown by the Chandal lab. Serine was implied to be important both from an extracellular standpoint as well as de novo synthesis by Rusty Jones's lab. One-carbon metabolism was highlighted by Marcia Hages. Methionine was shown relatively recently by the Control lab. And very early on, one of the really nice papers that showed that co-stimulation early during the activation here leads to upregulation of GLUT1, which is important for the acquiring this glycolytic phenotype that is important for the effector response. So during the effector response, the dominant pathway, so from this mixed bag during the first 36 to 48 hours, they become dominantly reliant on glucose metabolism. And it's been shown by many labs, including Erica's, that you need flux through glycolytic pathway to be able to produce these effector molecules. And there's multiple hypotheses why that would be, but it all boils down to the point that glucose metabolism is important here. So in the absence of glucose, these cells fail to produce robust interferon gamma and fail to fight a pathogen. Then when they differentiate, they lose this glycolytic driver, so they don't run the glucose metabolism by producing lactate anymore. They become memory cells and they become dependent on oxidative pathways. So it's fairly contentious whether this is dependent on CPT1A dependent FAO. We don't really know if that's the case or not, but they are oxidative. So this oxidative phenotype of the memory cells is really well described. There's a lot of mediators that drive this differentiation. Notably, fatty acid metabolism was shown by Erica's lab and by others. Other people have shown, like Rafi Ahmed, really nicely showed that mTOR is very important in the modulation of this. And these cells then become long-lived. So one of the phenotypes that we showed in Erica's lab is this is also associated with the formation of extensive mitochondrial fusion and tight cristae structures of the inner mitochondrial membranes. And all of these phenotypes drive this rapid response. So if you mess with your mitochondrial metabolism in the differentiation or after differentiation of memory cells, they fail to respond and mount a secondary resolution. Important to mention is even though they differentiate here and change their metabolic phenotype completely, they are very rapidly able to re-engage the secondary effector phenotype, which is very similar to the original effectors, again, dominated by this glycolytic phenotype and OXPHOS. So what this kind of highlights, and I think is very important for this talk, is that this requires metabolic remodeling and plasticity, both. So if you don't have the metabolic remodeling and plasticity, none of this can happen. So this requires changes in metabolic flux, as indicated by glycolytic flux, by OXPHOS changes. And we'll get to that in a little bit, what we mean with metabolic flux. And we're interested, like Erica's lab is, in how these changes are regulated. So there's a lot known and a lot of work going into the early activation, into the effector response, but these dynamic changes are still of interest. So how can you provide metabolic flexibility? And some people have already touched on this in these talks over the last 2 weeks. There are instant metabolic changes during differentiation, otherwise you won't be able to get these switches of phenotype. So their function is really tightly linked to their metabolic phenotype. Gene expression changes happen very early after TCR signals, as indicated in some of those papers that I highlighted, where enzymes are upregulated, transporters are upregulated. So then the question becomes, are these transcriptional signals just simply remembered and or imprinted or epigenetically modified so these cells can keep responding? But one of the, actually 2 strong papers published by the Cantrell lab and by Hongbo Chi's lab at St. Jude show limited overlap between the transcriptome and the phosphoproteome after activation and later on, suggesting that there's much more to it than simply transcriptional signals. So then the first question we need to ask, I think, and what I need to show you is whether metabolism is actually altered quickly during differentiation. And for this, I need to say we can modulate these pathways, as I just highlighted in the previous slide, to make an effector and a memory response with cytokine modulations in culture. So what we can do is we can make effector cells and grow them in the presence of IL-2, which become this dominant glycolytic phenotype. But we could switch them for 24 hours into IL-15, which is a different cytokine, which drives memory differentiation. The total memory differentiation in a culture dish takes about 3 days. So this is very early on after swapping these cytokines. And we extracted polymetabolites and sent it to General Metabolics. So I'll give them a shout out here. So Ed Driggers, CEO of General Metabolics, helped us do this. And they plot their metabolite pools that they measure in a whole cell extract as differentially expressed and visualized as using IPath3 from the EMBL. This is a really helpful tool for people that want to visualize changes in pathways. And every dot here is a metabolite. The color dictates fold change, and the size of the dot signifies the p-value. So you can see there's a lot of changes here. So given that I haven't shown this map before, it's important to highlight that these are the pathways we just talked about. Right here, we have glycolysis in red, the TCA in blue, and the PPP in purple right here. Now, if you see what changes, yes, there are changes in central carbon metabolism in the glucose pathway. So this goes down, as you would expect, because you go from glycolytic to oxidative. This is mirrored by an increase in pole sizes in the TCA. And we see a little bit of a change here in the pentoses. So there's a little bit more pentoses present in these memory cells. But however, all of this stuff also changes. So when we're talking about the pathways today, like looking at glycolysis, the TCA, the PPP, we're kind of ignoring all the other changes. So there's still a lot to be found, a lot to be learned about these pathways that we really don't understand yet. And my lab is really interested in understanding better whether these are regulatory metabolites and how this functions. So this clearly shows that there is a massive change in the metabolome. So just based on the pool sizes of metabolites, when you go from an effector response to a memory-like differentiation very rapidly. But it's important to point out here, when you do either expression analysis on the RNA level or Western blots for the proteins, a lot of central carbon metabolism enzymes are not expressed differently. So when you go from a glycolytic to a more oxidative phenotype, you don't lose, for instance, your whole glycolytic machinery. There's no changes in everybody's favorite loading control like GAPDH. That expression level stays high. So the enzymes are there, they're just not active. So that's a very interesting thing to think about. So what we set out to do is to look at, if you now take these cells and instead of giving them a different cytokine signal to drive a different phenotype, what if we take that dominant fuel away? So now you have an effector cell that are glycolytic, they're fully differentiated. Now we just deplete them of glucose and test how flexible their metabolism is, given that all these enzymes are there anyway. So the way we measured the first metabolic experiments are with the seahorse from Agilent, which is the extracellular flux analyzer. For people that are not familiar with this machine, this is actually their logo. Why they chose a seahorse, we still never figured that out, actually. It measures 2 parameters simultaneously in live cells in a monolayer in a plate. The 2 parameters are OCR, OCAR, which is oxygen consumption rate, which is a readout for what happens in complex 4 of the electron transport chain, and ECAR, or extracellular acidification rate, which is a proxy for lactate, which is being secreted here. So by measuring these two parameters at the same time, you get an idea of how much lactate is being secreted by these cells as a function of glycolysis, and how much oxygen is being consumed as a function of the activity of the electron transport chain. So when we do these experiments, it's a really simple setup. So you take your naive CD8 T cells, you activate them in a dish, so you mimic an infection. So you have a danger signal or co-stim, and you have a T cell receptor signal with CD3, which is not important. In the context of this talk, you expand them for 48 hours, then wait another 24 hours, so you remove the stimulation after this, and this is all done in 10 millimolar glucose media. Some people use IMDM, which is actually 25 millimolar glucose in their T cell experiments. We normally use 10 or 11 with RPMI. And I just put the reference values down here. For this audience, it's important to highlight this again, that we're probably in super physiological conditions in all cases in these cultures. And then after this, so they're fully differentiated at this point, we then put them in a 20-hour culture with either 10 millimolar glucose, so unperturbed, 3 millimolar, or 1 millimolar. And for the context of this talk, the 10 and the 1 are the important ones that we'll be talking about. And then we measure basal metabolism. So what we can see, and this is absolutely not surprising, because we reduce the glucose, and seeing that ECAR is a readout for lactate production from glycolysis is perhaps very unsurprising that ECAR goes down. But at the same time, we saw a slight increase, and it was significant, in the oxygen consumption in these cells. So this would suggest that these cells switch their metabolism from a dominantly glycolytic phenotype, although their mitochondria are active here, right? So I want to point out that they are consuming oxygen, and they're fermenting glucose to lactate. They do switch. So they downregulate ECAR, they upregulate OCAR. So this is just a proxy for the pathways, and this might not be reflective of the whole metabolome. So then we decided to look at the interconnected metabolome. So doing an assay in which we measure the metabolite pools in the cell. So this is what Joy really pioneered in the lab. So we did, again, the same experiment here. So we activated them in a dish. We expanded them in 10 millimolar glucose and did a dilution series of glucose overnight. And so we did a 10 millimolar versus a 1 millimolar, extracted metabolites, and then just did untargeted metabolite profiling. So this was done by Joy. And not surprisingly, just like the ECAR going down, we see a reduction in lactate and a reduction in glucose, which are highlighted here in orange in the 1 millimolar. So when you deplete glucose, you lose lactate. Entirely unsurprising. So when Joy then did a CAG pathway analysis, we found that the pathways that were down modulated, or the metabolite pools that were down modulated on this side of the curve in the 1 millimolar, were confined to amino sugar and nucleotide sugar metabolism, which all branch off of this part of glycolysis in a different direction here. So perhaps still not unsurprising. Galactose metabolism was hit, carbon metabolism, but we also saw purine metabolism, and of course glycolysis. So purine metabolism is part of the PPP here. So if you run the pentose phosphate pathway, you can synthesize purine. So those pools were going down too, suggesting that the loss of glucose leads to a loss of metabolites in glycolysis and in the PPP. But we didn't see many flags on this analysis of the TCA cycle. So then the question becomes, like, what are they doing with the remaining glucose carbon? So they have a little bit of glucose in the beginning, that 1 millimolar. So what are they doing with that? Is that changing? Are they making a choice because they realize they're running out of glucose? Because there are sensors for this, for those in these cells. So for that, we did the same experiment, but in this case, we incubated the cells with a dilution series of 13C, so heavy labeled, stable isotope labeled, universally labeled glucose. And in that way, you can actually follow where these carbons go. So I've highlighted them here as open circles, which here is the 13C. So this is the heavy labeled stable isotope part of glucose. The 12C is the remaining carbon that was already present before we started those cultures. So focus on the open bars for this, and that's what the statistics has done on. So what we noticed very, very clearly after 20 hours in glucose, the intracellular concentration of newly acquired glucose, so to say, is highest in the 10 millimolar and drops off very rapidly to the 1 millimolar. So this we don't detect anymore in the 1 millimolar, not surprising. The first step of glycolysis to get the glucose 6-phosphate, you see the same drop off. So you have a ton of glucose 6-phosphate in these 10 millimolar cells that have ample glucose present, and this drops off rapidly. But then they have a choice. As we talked about earlier, your glucose carbons can be made into pyruvate, which is depicted here. There's a bunch of enzyme steps here in between. And then they have a choice. They can go to lactate, which is kind of wasteful because you're losing these carbons, although you do regenerate redox cofactors, which will become important in a minute. or you can shunt it into the mitochondria. So you make mitochondrial pyruvate, which then assembles into the TCA, and are depicted here as 2 of the carbons from glucose being assimilated into citrate and succinate, are the 2 measurements we have. And when we measured these differences, we noticed that there's a drop-off in the intracellular amount of lactate in these cells. So they're stopping to make lactate, although the 3 millimolar, given that they have less pools of the upper intermediates, is still able to. The 1 millimolar drastically reduces this. And surprisingly, there was no significant difference in the amount of glucose that is now being used from up here to go this way to citrate. So independent of the glucose concentration, the assimilation in citrate seems similar, and the same is true for succinate. And it's important to mention, I mentioned glutamine earlier. I've cut that off of the figure here to make space. We did not see changes in glutamine metabolism in these cells. So the use of glutamine to the TCA in any of these intermediates is the same in all the glucose concentrations, suggesting that there's no compensation by glutamine to make these intermediates here to sustain this pathway. And that's also kind of reflected by the fact that there is no change in glucose. So this is also reflected, of course, in the OCAR to ECAR ratio. So if you look at the relative amount of lactate versus oxygen consumption, this goes up. So they use more oxygen consumption, and this is also reflected in the glucose allocation. So this fits nicely. And this would also suggest that although these cells are running out of their dominant biofuel for these pathways, they might not suffer so much bioenergetically, since they can still run oxygen consumption rate. And when you measure the ATP-AMP ratio in these cells, it's not significant. So we call this, we kind of say that there's no change here. So there's no loss of the ability to make ATP. So this suggests that this transient glucose restriction supports energy homeostasis, given that we still have the TCA running. And then the question, of course, immediately becomes, but what about AMPK, which has been shown to be a super important glucose sensor in T cells? So this was done by multiple labs, including the Cantrell lab and the Jones lab, which showed that upon glucose depletion, you activate AMPK and you dampen this pathway and increase oxygen metabolism. And actually, the Jones lab showed that this was glutamine dependent. So when we looked for AMPK, it's kind of shocking. 20 hours after TGR, we actually saw a down modulation of phosphorylated AMPK in these cells. So instead of this being activated, the phospho-AMPK threonine 172 is an activation marker, it goes down. So even though these cells don't have the dominant biofuel and AMPK was shown to be activated, this led to a downregulation in these cells. So this led us to wonder, because this is kind of counterintuitive with other studies and doesn't really agree very well. And we also looked at targets of these pathways like ACC1 and others, and they all go down. So it really looks like functional AMPK is actually downregulated by glucose starvation long-term. So we decided to do a time course for this to really address what's happening here. And we measured extracellular glucose and extracellular lactate on the bioanalyzer from Roche, in which you can use a little bit of biofluids, so in this case media, and measure the concentration of glucose and lactate at the same time. And what you can see in the 10 millimolar, these cells gradually deplete their glucose. So they start at 10 or 11. There's a little bit in the serum. We don't use dialyzed serum for this. We see a down modulation here. So they consume about 7 or so millimoles of, 4 millimolar of glucose and generate about 8 millimolars of lactate over time. So there's a 1-to-2 ratio, suggesting that most of their glucose is being used to be secreted to lactate. And when you look at the 1 millimolar, they start a little bit over 1, and you can see here at the 10 hour, there's a little bit of glucose left, but at 12 hours it's depleted, and this line becomes flat. So you don't detect glucose in the media anymore. And this is mirrored by the lack of extra lactate production here. So when we now look at phospho-AMPK, and I've just put the quantification in here over time, you can see that we don't see AMPK activation until these cells run out of glucose. So right as you would predict from the data from the Jones and the Cantrell labs, right as these cells run out of glucose, they activate AMPK, they upregulate it, their targets are also modulated. But somewhat surprisingly, even though there's no glucose present anymore, by 18 and 20 hours, they resolve this and they lose this phosphorylation and it's dampened again. So perhaps this is the moment that these cells are metabolically remodeling, and this might well be dependent on AMPK signaling as shown by other people. I must say there's a spike in glutamine consumption right around the 12-hour time point, so there might be a role for glutamine at this point. But when you look at the end stage, there's really no indication that glutamine metabolism is altered. So this suggests still that AMPK signaling is activated upon glucose depletion, But after 20 hours, the signal is back to baseline. And if anything, it suggests that it's a little bit lower. So what is happening here? How are these metabolic changes mediated? How do these cells know they run out of glucose? And how do they resolve this? And is there a change in carbon allocation? Is that what's happening? So for that, we chose to look at redox changes. And I'll highlight why we did this in a very short fashion. So I mentioned earlier, glucose can be made into pyruvate with a net gain of 2 ATPs. Important to mention here is that you have an NAD plus consuming point here at GAPDH, which is then recovered by LDH in the production of lactate. So these enzymes are tightly linked in function because they recycle redox couples to keep the pathway running fast. So this is suggestive that if you were to change the levels of either of these redox cofactors, that the pathway might either stop here or be directionally influenced, so to say. And the same is true for this redox couple here. If you run out of NADP, you might not be able to run this pathway to the pentose phosphates. And this has multiple consequences downstream, but it would also change the ability of using these carbons. So this is what we set out to do. So for this, so again, so if you lose this here by not producing lactate, you might lose the ability to run glycolysis through here. So if you measure this now as the NAD-NADH ratio, we didn't see any significant differences. So suggesting that even though this reaction doesn't happen anymore, this is not perturbing the ratio of NAD to NADH. And this is a whole cell isolate. So I also want to point out that there are a plethora of other reactions that use NAD and NADH, including the TCA cycle, but there are many, many more. So this might not be reflective very well of what is happening here, so that we failed to show any differences here. And then, as I mentioned earlier, if you lose NADPH, you won't be able to revert your oxidized glutathione back to reduced glutathione, and therefore lose your GSH. So the ratio of these two would alter, and that we did see. So the GSH to GSSG ratio swapped to a more oxidized state. So this is suggestive that in these cells, at least this is being lost, leading to an accumulation of GSSG. And we actually showed in data that I don't have time to show here that this is not dependent on de novo glutathione synthesis. That is completely conserved here and is depending on glutamine. So this is really, we think, to do with the recycling based on the redox couples. But as I mentioned, these pathways are happening in the cytosol and in the mitochondria. So how do we resolve this? Is this really just as simple as the pathways changing direction, or is there a compartmentalized redox issue? And as was shown really nicely by Sophie a couple of weeks ago, you can do subcellular fractionation and subcellular metabolomics in cells that she showed, but for T cells, this is really not trivial. So although they really nicely showed acyl-CoA metabolism in different compartments, we failed to do this in T cells. It's like these cells are ping pong balls. You either burst them completely or you don't burst them at all. So we get leaking compartments, we don't know the stability of the redox couple. So this was a bit of a problem. The dynamics became even harder to track. So we really wanted to look over time what happened to these redox couples. And this was not easily done in many samples because yeah, this accumulates into a lot of work. So we decided to use a redox sensitive Rho GFP. So this is a mutated eGFP, which has cysteines introduced that make it redox sensitive. And the details are really nicely outlined in this paper in Circulation Research from 2010. That's where we got the vectors from through AdGene. And so the trick here is that reduced GFP normally excites at 488 and emits at 515. So this is normal GFP. But when it's now oxidized based on these newly introduced cysteines, it becomes sensitive to the 405 laser. So this changes the excitation spectrum, and you can measure those changes. So the way we did this is I cloned the constructs that we got from these people from AdGene into an MSCV vector backbone. So the normal Rho GFP, which is all over the cell or in the cytosol, and the mitochondrially targeted Rho GFP that we got out of this paper, and then infected cells, and did the same experiment. So we infected these into the primary T cells. So again, activated the normal way, did a glucose dilution series overnight, and then tried to image this. So I'm just showing this image as an example. So you can see that the normal Rho GFP is all over the cells. You can see it in the cytosol and the nucleus is everywhere. So we mostly see it cytosolic in these, and you can measure the reduced and the oxidized version of this GFP. And of course, it's dependent on the level of expression. And this is the mitochondrially targeted one, which becomes really nicely punctate, as we showed in 2 independent papers in Cell from Erica's lab. These effector cells have punctate mitochondria. You can nicely see that here. But now you have to start drawing manual ROIs to measure this. Given that these are so nicely divergent, I've decided to do this by flow, which is much faster, and then compute the ratios of reduced versus oxidized GFP. So we use control GFP, just regular unmutated GFP as a control, and then subjected them to this glucose starvation overnight. So either in 10 or 1 millimolar, you can see there's no severe change here. But when you look at the cytosol, so at the regular row GFP, you see a massive oxidation of the cytosol based on glucose starvation. And we see the same in the mitochondria, albeit less drastic. But this could be the dynamic range of the detection of the row GFP. So we have 10 more millimolar. So this is just reflecting more reduced and more oxidized. So both of them become more oxidized. So the first question is, if we're so much more oxidized and we're losing some of our glutathione, do we get ROS? And we failed to detect an increase in cellular ROS, and there was no significant change in mitochondrial ROS either. So this suggests that although these compartments are being more oxidized as a global effect, there was no accumulation of ROS in these cells. So that this might be reflective of the fact that we still had some GSH left to buffer this. So then the question becomes, does this alter substrate handling now? So we know that these compartments are more oxidized. Is this a driver of different substrate allocation when you re-expose these cells to substrate? So we go back to the same experiment we talked about earlier. So we do a dilution series of glucose overnight. But now instead of just measuring the pool sizes, we pulse these cells with 10 millimolar 13C glucose. So again, heavy labeled, stable isotope labeled glucose. And after 5 minutes, we measure changes. So again, these are the open circles here that can be allocated in the directions of glycolysis, the pentose phosphate pathway, and the TCA, although it's 5 minutes. So we really only measured the glycolytic intermediates and the pentose phosphates. So when Joy did this analysis with CAG pathway hit analysis, we looked at the total pool changes and the isotope labeled allocation. So this looks both at the amount of the metabolite and the metabolite derived from new glucose in these cells. And we found, not surprisingly, carbon metabolism is reflected in both, but we found mostly the pentose phosphate pathway being driven up in the previously starved cells, suggesting that these cells allocate more carbon from new glucose into the pentose phosphate pathway. And this is also highlighted by pyrimidine metabolism. So both the isotopes and the pools go up. And in line with this, if you measure the redox, so again, with these redox sensors in the cytosol, you can see that as you add glucose back in, you can see these cells become more reduced again. So this is the more reduced side. So these were the start cells that were more oxidized and then now become more reduced, suggesting that there's a global change in the redox factors over time in the cytosol during this 5-hour time point, suggesting that if these carbons are going this way, they might be regenerating NADPH, and that could be in part reflecting this change in redox factors. And the same might be happening here by generating more NADH. So we measured this in the GSH and GSSG ratios. And what you could see is that although it was still down in the 1 millimolar after a glucose refeed, again, after 5 minutes, so this is 300 seconds, we see that it's no longer significant and it does come back up, suggesting that some of these carbons being pushed here allow you to run this reaction as well, to push GSSG back to GSH. So this is at least in line. But this doesn't tell us, so just measuring these static pool sizes and isotope labeling doesn't tell us the dynamics, which is clearly indicated here that this is not a one-off hit, it is actually dynamically changing. So we also did a 30 and a 90 second isolation and then measured the specific allocation over time from 30, 90, and 300 seconds. So this was done together with Annette in the lab, and I'll highlight Annette later. She's now my lab manager, which is also my wife, which is great to have somebody around that is willing to work with you on a day-to-day basis. But the quickest we really could do it was 30, 90 seconds. So what we did is we pulsed these cells with, again, the open bars here are the important ones, the 13C glucose. And what we could see is that when you look at glucose inside of the cells, the black bars being the present glucose, which is the closed bars, which is of course different in the two glucose concentrations. But if you look at the open bars now, you can see a slight increase. So there's new glucose import over the 5-minute time point in the control cells, but this is massively augmented in the previously starved cells. And although I'm not showing it here, these cells express more GLUT1 on the surface. So there might be something to do with the ability to bring more glucose in. Although GLUT3, which is the more efficient transporter, is expressed at the same level. So when we now trace these carbons through the pathway, we can see this difference is even stronger reflected in glucose 6-phosphate. So the first step, after the first ATP is consumed by hexokinase, you get glucose 6-phosphate. This is massively enhanced in these cells. So not only do they bring in more glucose, they also metabolize it quicker. So now the question becomes, are they perturbed, right? So this could be that the PPP is not working, or glycolysis is not working, and they're accumulating glucose 6-phosphate compared to these. But if you, I don't have the data here, but fructose 1,6-phosphate and the trioses are also increased in the previously starved cells. But now they have a choice. So they can be wasteful and make lactate and chuck it out of the cell, or they can start building and then make nucleotides and run an anabolic pathway. And we saw this. So not surprisingly, as shown in the KEGG pathway hits, the PPP was enhanced. This produces NADPH, and we see an increase in pentose phosphates over 5 minutes in the starved cells, suggesting that the allocation of these glucose carbons is not only faster, but it's changing. The direction is changing. And we saw no increase in lactate over time in these cells, at least not over the 5-minute time point, which is rather fast, but you could see it. So this suggests that NADPH should be generated. And we haven't got them as ratios here because there was no detectable NADPH in these cells. So computing a ratio is kind of hard. But what you can see is the NADPH level was lower in the starved cells, which is reverted 5 minutes after you give them glucose, suggesting that this pathway is now running because the accumulated NADP is actually going back down to control levels in the 1 millimolar after you refeed them, suggesting that this pathway is now activated. It allows you to recycle these redox intermediates and therefore likely get more reduced glutathione. So we also looked 6 hours after. As I said, this was a 5-minute pulse, so you can't really measure the end product nucleotides or other intermediates. And what we noticed is that we see an increase in newly synthesized ATP as a nucleotide readout. This was the same for AMP. We also see augmented serine production, so they make more serine from new glucose 6 hours after. And they still make less lactate. So although the amount of de novo lactate here is not changing, the pool size inside of the cell is not coming back to where it was, suggesting that these cells now have an ability to build more than to shed. And the GSH-GSSG ratio after 6 hours is completely balanced again, suggesting that this is now in homeostatic state again, likely due to the recovery of NADPH. So this suggests that your TGR-CD8 T cells are more anabolic, at least over this 6 hour time point. They can acquire new glucose, but they don't do what they did previously with this glucose. But does this matter? Why do we care? Why are we actually measuring these things, and does this matter for anything? So now we go into the functional side of things. So when you look at the CD8 T cell function in the tumor microenvironment, a lot of work has been done. And as shown by Alex Muir last week, or 2 weeks ago now, by Thales, the tumor microenvironment is completely different than the circulation. It's depleted of nutrients. And this is a review from the Rathma lab, which I really like, which highlights that this is depleted and there's altered nutrient availability. And specifically, glucose and glutamine, highlighted here, are competed for by tumors. So it was already shown by Isaac Harris, by Thales, that these are high glutamine consumers and glucose consumers, depending on the different transporter expression. Equally, T cells depend on these pathways. Like I said earlier, it's been highlighted a lot that glucose is needed for effector function, the same for glutamine. So if you dampen these pathways, you don't only dampen glycolysis, but you also dampen OXPHOS. So this leads to dysfunction in the T cells. Another part is the active immune suppression by PD-1 ligation, which is highlighted here, which leads to a dampening of mTOR signaling and pathways that are associated with MYC, with glycolysis, but also with OXPHOS. And accumulation of lactate over the top of that dampens effector function, as highlighted here, which is like termed waste accumulation, although some cells are able to use lactate as a biofuel. So like I said before, glycolysis and mitochondrial metabolism are perturbed. So how do we deal with this? So how can you enhance the anti-tumor CD8 T cell function? So I want to just highlight a couple of stories. We're not the first ones to mess with glucose metabolism in the setting of trying to make better anti-tumor T cells. So if you differentiate T cells from the get-go, so if you make these naive T cells biased towards a more memory-like phenotype, and you can do this by blunting glycolytic metabolism during activation with 2DG or AKT inhibitors for selecting for cells with lower membrane potential. You get this memory-like cell that works much better in anti-tumor therapy for adoptive cellular therapy. And this was really pioneered by the Stifo lab and the Gattinoni lab that both did these experiments, which was really nicely done. So this is done by 2 ways of pharmacological inhibition of glycolysis. On the other hand, if you enhance mitochondrial metabolism, this can enhance T cell function in the tumor microenvironment. So by enhancing mitochondrial mass, either by PGC1 alpha overexpression in a really nice study from the Del Goff lab, or with treatment of activators of the same pathway that can activate both FAO and mitochondrial biogenesis by bezafibrate, which was really nicely shown by the Honjo lab. We can increase the mitochondrial mass, and these cells are also more robust in the tumor microenvironment. Similarly, in Erica's lab, we used MDV1 previously, which leads to enhanced mitochondrial fusion. And we know that glucose starvation also leads to an increase in mitochondrial fusion and not necessarily mitochondrial mass, as we showed in our CD28 mitochondrial paper a few years back in Cell, also in Erica's lab. So we think that this TGR might do something similar and drive this mitochondrial phenotype because they have more ATP, they have more oxphos, less glycolysis. So maybe we're actually mimicking this. The bonus of our system is that we can massively expand these cells by activating them into a fast proliferating cell type, generating an enormous amount of these cells to treat, in this case, we treated mice with them. But this is a very powerful way to do it, and you only need 20 hours of one perturbation in the media. So you're not treating them with a drug like MDV1 or bezafibrate. We don't have to express anything. We literally just change the glucose. So we wanted to test whether this increase in mitochondrial biogenesis and anabolic metabolism would actually be beneficial. So to do this, we first tested this in a dish with spheroid cultures. So we made 3D spheroid cultures of B16 ova. So they express chicken ovalbumin, which is not necessarily the most natural thing for a melanoma cell line to express. But the nice thing about this ovalbumin expressing spheroid, we can target this with a specific T cell from the OT1 mice. So the OT1 mice respond to this peptide, which is part of chicken ovalbumin. So if you activate these cells, they all have the same T cell receptor with this peptide. You can then target them specifically to kill tumor cells that express this ovalbumin. So if you activate these cells in a dish, again, in normal glucose, expose them to 20 hours of differential glucose concentrations and put them in cultures with these spheroids, you can see here that we have day 0 T cells. So these are the T cells, these tiny things lying around here that are outside of the spheroid. If you wait a day, and this is what we did, so we incubate them for 20 hours, you can see they all crawl inside of this tumor and they start being activated because they recognize this ova. And we do this in the presence of IL-2, which these cells need to survive, and new glucose. So even though they were starved overnight, when we put them in this environment, we expose them back to all the normal conditions to equalize the playing field for these cells, which would happen in vivo as well. And to our delight, really, we saw that these orange ones here, so the previously starved cells, produced more interferon gamma, as measured here, by percentage and by MFI, which is quantified on the right. And they also had more granzyme B. This granzyme B and interferon gamma I highlighted in one of my early slides are important effector molecules to help kill tumor cells. We failed to demonstrate differential killing because these spheroid cultures are really hard to analyze. So we really wanted to look at this more functionally. So we know they have the ability to produce more effector molecules, but do they actually do something to a tumor? So for that, we went one step further. So we now know it works in vitro, but of course, that's easy to demonstrate. It's not as important as showing whether these cells work in vivo. So what we did is we did the same experiment. We used the same cells, but this time we used EG7 or EL4 ova, which is a melanoma cell line, not a melanoma. It's a lymphoma cell line, which you can inject in the flank of a tumor and then wait 8 days before you inject T cells. These are injected into wild-type B6 mice. So these cells have an immune system, but they fail to control this tumor. So we inject about 1 million cells. And then we set up the cultures in parallel, starve them again like we did before, and then infuse them into these mice. So the first thing we noticed is after we inject these cells, so what we did is we waited 8 days. So these tumors grow to a substantial size. So they're 8 millimeters in diameter, which is relatively big. and it's relatively late. Most of these assays we used to do at day 5, where the tumors are small, where you still struggle to control all of them. So we really pushed it late, and we gave them less cells than we normally would do. In a lot of other assays, we used 5 million cells. So we're really pushing the system here to see if these control cells, which were grown in normal glucose, versus the starved cells in 1 millimolar glucose would actually have a benefit. So the first thing that was very obvious is when we bled the mice 8 days after we give them these T cells, is that the number of donor-derived CD8 T cells of the total CD8s, remember these are wild-type mice, so they have their own CD8 compartment, was increased and significantly increased. And this was done in 3 independent experiments, and we constantly see this increase in circulating cells. And this is actually persistent over time. So even when you look much later, so 28 days later, you can still see this, that these TGR cells have a survival advantage in vivo or proliferation advantage. We haven't really deduced what this is, whether this is proliferation or simple survival. And if you take these cells at day 8 and you re-stimulate them in a dish, they also are able to produce more gamma. So some of them fail to produce gamma at all from the control mice, whereas these guys here produce ample gamma, and those significantly higher. But I think the important thing is here, and I actually didn't believe this at first. I had my master's student in the lab at the time, Nina Lichthart, who hopefully is watching as well, actually co-measure these tumors, because everybody was blinded to what was happening. So I want to make sure we didn't get this wrong. But you can see that these orange cells, so these cells here, exactly the same number, same antigen, same expression, cleared these tumors completely, whereas about half of the control cells failed to control this tumor. And unfortunately, we had to sacrifice these mice because the tumors became too big. So this suggests that this enhances anti-tumor function in vivo. And this is really nicely aligning with other studies from other labs that this augmented mitochondrial metabolism really can benefit these T cells in vivo. But now, to make it more relevant and kind of link it back to the work from the Hangzhou lab, who developed and actually got a Nobel Prize together with Jem Allison for checkpoint therapy, is to combine it with immune checkpoint therapy. So we're doing the same thing with the cells, and we're putting them into a different system now. Here we use the B16F10 over positive cells. So this is a melanoma tumor. This is really hard to treat with T cells. It's incredibly aggressive, and it's really hard to control the growth of these in, again, in wild-type mice. And we're injecting, again, these starved T cells, but we're now combining it with 3 injections of isotype control or anti-PD-L1 antibody at day 3, 6, and 9 post-tumor implant to benefit the T cells. So I highlighted that in the review from the Rathma lab, that PD-1 signaling can really mess with your T cell efficiency. So if you now ligate PD-L1 on the tumor with an anti-PD-L1 antibody, you open up the signaling from PD-1, you don't get this blunting signal anymore, and these cells benefit. So first I want to highlight, when we looked at these cells again, and we looked at the percentage of donor CD8 donor CD8s of total CD8s in circulation of these mice, again, starving them overnight of glucose benefited these cells greatly in the number of circulating cells. So this is very similar. And now we have a benefit. We can look inside of the tumor. So in the previous experiment, we were never able to look at how many cells will infiltrate the tumor because these tumors vanish so quickly. So in this setting, we don't clear the tumors completely, so we can isolate them. And this was done by Petya Apostolova and Erica's lab, who wrote the protocol and did all these experiments as we had already left for Canada. So I really want to commend her on this. This is a lot of work. And she actually counted the number of CD8 donor cells per milligram tumor tissue and showed that also here we have a massive increased number of previously starved cells inside of these tumors. So not only do they survive or proliferate or sustain better in circulation, they also get into the tumor better. And therefore, perhaps not surprising, since we have more T cells there that are antigen specific and they control the tumor somewhat better. And as I said, these tumors are super aggressive. They're not very easy to clear. And this is really stacked against the T cells to clear them. But we see a significant improvement of the tumor control here in the orange bars versus the black bars. And you can see that the host immune system really, really struggles here. So if you now combine this with checkpoint antibodies, so anti-PD-L1, so this regimen here, and you now look at the circulating T cells, we still see an increase in circulating T cells from the donor in the TGR cells. So this benefit is independent of checkpoint therapy. However, we now see that the control cells are able to invade this tumor better. So you go from a highly significant difference here, or like a rather significant difference here, to a much smaller difference here, benefiting these control cells to get into the tumor. And this was actually shown by the Honjo lab with bezafibrate and other activators of mitochondrial metabolism. If you activate the mitochondrial metabolism, these cells get into the tumor better and they function better. So if you now look, not surprisingly, there is now no difference anymore. So although early on it looked like there was a little bit of a difference in the tumor control, these now both control the tumor equally, suggesting that that has to do with the function of anti-PD-L1 to drive these cells into the tumor, even though we don't see an increased number of circulating control cells. So what we think is happening here is that the increase in mitochondrial activity induced by anti-PD-L1 in the control cells can no further augment our starved cells because they already have the perfect mitochondrial metabolism. So that's one of the postulations we think that this mechanism might be occurring. So TGR enhances anti-tumor function in vivo, although here it's very modest, but it is giving them a benefit. But does not further augment anti-PD-L1 therapy, likely through a mitochondrial pathway that's already maximally engaged in the starved cells. So as a summary, I think I've shown you for this part of the talk that metabolic remodeling is crucial for T cell function, as highlighted by a lot of other studies, not necessarily just ours. And I just made this in general cellular, but in this case, T cell metabolism is influenced by nutrient availability, as we can see by just taking away one dominant nutrient. Transporter expression, so if you don't have glucose transportability, it's really hard to use it, but you can't get it into the cell. Our starved cells express a bit more GLUT1, so that might be one of the reasons they do better. Enzyme expression with a question mark. So there are enzymes that are differentially expressed during these different pathways, but the ones we were looking at, so G6PD-H, GPI, GPAT, all of those seem to be equally expressed in our cells by both mass spec and Western blots in our lab in Vancouver. We definitely have an influence of enzyme cofactors, such as redox couples. Whether this is the driver, so actually the chicken or the egg, we don't know, but it correlates really nicely. We think this is to do with nutrient signaling, nutrient sensing signaling pathways, which my lab is very interested in flushing out in more detail. So we're really setting up to start that work. And metabolic enzyme activity. So if you change the metabolic enzyme activity, not necessarily expression, be it by localization, by cofactor availability, this could be driving a lot of these phenotypes. And I think how carbon flux is regulated in these cells remains incompletely understood. I think we have a little bit of a better idea, and we open up a couple more doors here, but it's definitely of therapeutic importance for cellular immunotherapy of cancers, and especially for in vitro generated T cells. So this brings me to the thank you slide for the Max Planck. So first highlight Erica. Again, if you want a good supervisor as a student or a postdoc, contact her. She's fantastic. She's been a big driver of my career for sure, and she's been very supportive even since we've left. Joy, right here, side by side, like on the paper. She's an absolute wizard with the mass spec. She's really changed the Pierce lab of looking at metabolism for sure, together with the Metabolomics core facility. Jörg, shout out to him. And then Dave, who has recently also left Erica's lab and has gone back to his home country of New Zealand, who we used to call the Pierce Lab Wikipedia. It's unbelievable. If he goes, I think I remember, he's generally right. And then I highlighted Pecha, who really jumped in for the rebuttal experiments and pioneered these extra in vivo experiments. So thank you to them. And then I want to take a couple of minutes. So I know that I'm going a bit long here. So just a couple of minutes to introduce what we're doing in Vancouver. As you can see in the forest there in Vancouver, it's quite nice. We have these little critters running around. It's been an epic place to live. It's absolutely fantastic. First, we still continue to work on unraveling the complex interplay of immune cell metabolism and function. So this hasn't changed from what we did in Erica, but I think we have a slightly different approach. Hopefully not too competitive, because I really don't want to compete with the powerhouse of Erica's lab. And just to highlight this, we are looking for a curious and talented graduate student. So if you're interested, please email me with your CV, and we can talk about options in the lab. So we have two funded projects in the lab right now. One is metabolic conditioning of expanded primary human T cells to really extend this to antiviral and anti-cancer T cells for adoptive cellular therapy. And this is co-PI'd by Dr. Suzanne Verkouteren in the clinic at BC Children's, who is an expert in leukemia therapy and antiviral therapy. This was funded by the BC Children's Hospital Foundation and the Childhood Diseases team at BCCHR. So we got a small pot of money for this. We have an undergrad coming that I'm very excited about from Toronto, who is supposed to start this summer. So Fingers crossed the lab opens in a couple of weeks so we can do this work. And a second project is the cell intrinsic regulation of central carbon metabolism in effector T cells. So really linking nutrient availability, nutrient signaling to what these cells do with carbons. And we just found out yesterday that this was funded by NSERC in a discovery grant competition. So that's why we're looking for students. So hit me up if you're interested. So what we want to do is we want to identify signals that control substrate usage. So as I highlighted in the beginning of the talk, the energy versus biomass. So how do these cells decide? So this is the same picture I've showed a few times now with all these different pathways being driven in the different cell types. But it's important to note that not all cells use all nutrients available. And that's not necessarily just by expression of transporters, because effector cells use lots of glucose to make lactate. Yet memory cells use less glucose and decide to make fats with it, but they still use some glucose. So this is already showing you that there are changes here. This might be expression driven, but that can't be the whole story. So the glycolytic machinery is present in both. So as highlighted by the Matt Hentz lab and by Erica's paper in Cell, glycolytic enzymes can also bind RNA and change function of other proteins. So this is a really important principle to remember. But during expansion, T cells duplicate about every 8 hours. And this is probably an underestimate, if anything. And for this, they need to make DNA, RNA, protein membranes. It all needs to be made new to rapidly sustain this proliferative capacity here. So as I highlighted before, glucose is one of the nutrients. As highlighted by other speakers in this seminar series, there's a whole lot of other fuels that are used for this. But a really important thing to note is that in vitro is definitely not the same as in vivo, because as highlighted by Alex Muir, by Thales, by studies from the Cantor lab, we need better media. So we need to be better able to model the in vivo situation with the in vitro situation. And one of the ways to do this was really nicely demonstrated in mice by the Jones lab, who just published a paper late last year in Immunity, which we just did in our journal club, that showed that in vivo activation, these cells become more anabolic than they would do in vitro. And that could also be one of the reasons why our starved T cells do better in vivo, because they're more like in vivo activated T cells. So that's one of the things. So I go back to the poster here. So one of the things we're interested in is how glucose is being used to make lactate, but also to make nucleotides or lipids. So this is spearheaded by a super talented grad student in the lab who started in December, Ju He, in support by one of the undergrads from UBC biochemistry. These students are amazing. Like, they're so good. I would have never made it into my own lab if I see what I can hire right now. So they together have been looking at nucleotide and lipid synthesis. And then we have another project running in the lab that is co-supervised by Dr. Bojana Rakic from the clinic. She's a biomedical geneticist. And together with a postdoc from Megan Leving's lab, we're supervising another undergrad student, also from UBC Biochemistry, Serena. And they're looking at lipid metabolism in T cells as well. So those projects are up and running, and we hope to go back to the lab soon to be able to finish them. And that brings me to my last slide of the lab as it is. So here's Serena, Ju He, and Cassandra, Kirsten from the Leving's lab, Annette, my lab manager, and significantly better half. Me, you can't see the view. You can see the mountains from the lab. So you can actually see the North Shore Mountains in Vancouver. So it's a really nice view to have if you're interested. So I've highlighted these people. I've highlighted the funding agencies. So I'll leave it there. And I hope to see you all interacting on Twitter. And thank you so much for, again, for the opportunity to present my work, Philip, for the White Magara Lab. And see you guys out there. Thank you.